Hi everyone and welcome to my presentation. Let me thank all of you, those who have subscribed to my channel, and I will be making many more of the subject videos useful for your clear understanding and preparation for your examinations. Let me introduce myself to you all. I am Dr. Srivani with 30 years of teaching experience, an associate professor and head department of zoology at Sarojini Naidu Vanitha Mahavidyalaya. As you can see on the screen, my presentation is about protein synthesis. This topic is relevant to all the students of 12th, UG and PG. So, hope this presentation will be useful for you all. Let me start with it. So, first let us try to understand what are proteins. Before going to the synthesis of them, let's have a basic idea of as to what are proteins. Yes, proteins are giant molecules of utmost biological importance because these not only form the building blocks of the cell but act as enzymes controlling all biochemical reactions going on in the living system. As you all already know, proteins are made up of amino acids which are bound by a peptide bond between them. So, they are chemically the polymers of amino acids. And all the proteins which are required by the cell are synthesized by the cell itself. Protein synthesis therefore includes arrangement of a definite number of amino acids in the polypeptide chain in a sequential order. This sequence is determined by the sequence of nucleotides in the polypeptide chain of DNA. So, a sequence of 3 nitrogenous bases which are referred to as the genetic code or the triplet code. Okay, codes for one specific amino acid of the polypeptide chain. Now, coming to the central dogma of protein synthesis. This central dogma was coined by Francis Crick, according to whom the flow of genetic information happens in one direction only, that is from DNA to RNA to the synthesis of the protein, and DNA here replicates and new copies of DNA are formed. During the process of transcription, DNA is transcribed into RNA, and this RNA is translated into the proteins. So, central dogma means once the information has passed into protein, it cannot get out again. The transfer of information from DNA to RNA or from RNA to protein may be possible, but transfer from protein to protein or from protein to nucleic acid is not possible. The two processes involved in the protein synthesis or the central dogma is the transcription and the translation. As shown in the previous picture also, that from the DNA by transcription RNA is formed, and from the RNA by translation proteins are synthesized. So, let us dwell into the mechanism of protein synthesis. So, the first step involves transcription of mRNA from the DNA. So, in the presence of DNA dependent RNA polymerase enzyme, the genetic message is encoded or transcribed into mRNA, the messenger RNA. Then the two strands of the specific DNA molecule uncoil, and one of these two strands acts as a template from which the mRNA molecule will be formed with the complementary nucleotides. In case of RNA, in place of thymine, as you all already know, uracil is present. So, the base pairing will be A to U and G to C. The mRNA makes its way through the pores in the nuclear membrane through the cytoplasm. So here it forms a complex with a group of ribosomes termed as polyribosomes. And the synthesis of mRNA, as you can see in the diagrammatic representation, happens from 5' to 3' end. So the transcription of mRNA from DNA, in case of prokaryotes, it is only single RNA polymerase enzyme is present, which catalyzes the synthesis of different types of mRNA. So RNA polymerase consists of 5 different polypeptide chains, 2 alpha, 1 beta, 1 beta 1 chain, 1 omega, and 1 sigma factor is also present, which is the initiation factor to the RNA polymerase. The sigma factor chain is loosely attached to the remaining polypeptide chains, which constitute the core enzyme. So the process of the formation of the mRNA from the DNA, that is the transcription process, involves 3 steps: the initiation, elongation, and termination of the chain. So the first step is the initiation step. So the sigma factor helps in the recognition of the start signal on DNA molecule and directs RNA polymerase in selecting the initiation site and directs the binding of the enzyme to the promoter region. The binding of RNA polymer to DNA involves the beta subunit. RNA polymerase initially binds to an RNA polymerase recognition site on the DNA promoter region. It then diffuses to a binding site consisting of a sequence of 7 bases TA, TAA, TG, called as TATA Tata box. You can see here in the diagrammatic representation the sigma factor identifying the promoter region in the part of the RNA polymerase enzyme, and the region identified as TATA region. Then the next step in the transcription process is the elongation process. After initiation, the chain has to grow, so the next step will be the elongation process. With the binding of RNA polymerase to DNA, unbinding of the DNA double helix happens, so that a short segment of DNA molecule opens up. The chain elongation takes place by the addition of activated GTP, ATP, CTP, and UTP to one strand of DNA template, for which for each of the nucleotide is added to the growing RNA chain. A pyrophosphate is released, which will be rapidly hydrolyzed to inorganic phosphate. So the elongation of the mRNA chain takes place with the help of the core enzyme, which moves along the DNA template in the 5' to the 3' direction. So the next step in the formation of the mRNA is once the initiation happened and then the elongation of the mRNA chain is completed, then the termination process happens. So during the termination process, after the chain elongation process is completed, the sigma factor dissociates from the core enzyme. Since the sigma factor is the initiation factor for transcription of mRNA from DNA, it can be used by another polymerase molecule and start the initiation process for mRNA synthesis. And another additional protein called as a Rho factor brings about the termination of the RNA chain. The factor is a tetramer molecule which probably binds to the RNA polymerase enzyme. Transcription of mRNA, in case of eukaryotes, there are at least 2 types of RNA polymerases. Whereas in the previous slides I have explained that in case of prokaryotes there is only a single RNA polymerase, but in case of eukaryotes there are two types of RNA polymerases. The first one that is present in the nucleus is RNA polymerase A, the one which is responsible for rRNA synthesis, and the second type of RNA polymerase is located in the nucleoplasm and is represented by RNA polymerase B. It brings about the synthesis of heterogeneous nuclear RNA from DNA. A sequence of about 200 nucleotides of adenylic acid poly A gets attached to the 3' end of hnRNA with the simultaneous disintegration of 5' end, resulting in the formation of poly-AmRNA. It diffuses out of the nucleus into the cytoplasm where it participates in protein synthesis. The 5' end of mRNA molecule possesses 7-methyl guanosine, while 3' end terminates in poly-A sequence. Therefore, mRNA molecules are said to have marked ends. With the formation of mRNA from the DNA template in the nucleus and the movement of the mRNA out of the nucleus through the small pores in the nuclear membrane marks the end of transcription process. With this, we come to the end of the first step in the process of protein synthesis, that is transcription of mRNA from DNA. Do follow me in my next video for the process of translation to complete the protein synthesis. Thank you for watching. Do not forget to subscribe to my channel.